Thank everybody who came here today, all of our guest speakers. Thank you so much. We are thrilled to have you all here for this fantastic day, which has just been really exciting from my perspective. And thanks to all of the members of the audience, too. We had over 100 people register, which in my opinion is fantastic, from all aspects of Mount Sinai community, researchers, scientists, and clinicians as well. So thank you very much for coming. I'm going to talk about the neurodegenerative tauopathies a bit and start with a disclosure. In the interest of full disclosure, I am engaged in a research collaboration with Genentech. The neurofibrillary tangles of Alzheimer were first described by Alzheimer in 1906 when he applied a new stain, the Bielczewski silver stain, new at that time, to the brain of a woman that he performed an autopsy on who had an early onset dementing illness. And these are some drawings from one of his original papers showing the classic flame shape of these abnormal aggregates that are appearing inside neurons. This neuropathology forms one half of the neuropathological hallmarks for Alzheimer's disease. From this day on, well, there wasn't that much interest in his work until many decades later, but now there is a massive race to understand how and why these abnormal aggregates of the tau protein, we now know it's the tau protein, how they contribute to neurodegeneration. So the neurofibrillary tangle is composed of the microtubule-associated protein tau. Tau normally is bound to the microtubule, and its normal function is to stabilize the microtubule, which serves as a track for cellular transport down axons. Tau probably has other functions as well, including neurite outgrowth and signal transduction. In the context of neurodegeneration, we observe tau dissociated from the microtubules, abnormally hyperphosphorylated and aggregated into these clumps. How these post-translational modifications occur, we don't know for sure, and what are the implications of these changes. One of the things that I've always found kind of surprising is that we're not certain whether or not these aggregates cause a loss of function or a gain of function. So it's quite plausible that these aggregates are toxic and have some effects that way, and it's also plausible that these aggregates sequester tau away from the microtubule and prevent it from its normal function. So today, I'm going to talk not about all the tauopathies, because there are quite a few. We're going to focus in on a couple, but the purpose of this slide is to show you that tau aggregates occur in a lot of different contexts, clinically and neuropathologically. We see tangles in over 23 different diseases, depending on whether or not you're a lumper or a splitter. So clinically, you can see neurofibrillary tangles in cognitive disorders, dementias. You can see them in movement disorders and motor neuron diseases. You can also see them in the context of certain psychiatric symptoms, suicidality and depression and chronic traumatic encephalopathy, for example. Neuropathologically, when you look at the brains of patients with tauopathy, you can find these aggregates in neurons, but you can also find them inside of glia, astrocytes and oligodendrocytes, and we can use this information to help guide the diagnosis. We also use the regional distribution of the abnormal tau to help guide the diagnosis, and that regional distribution contributes to the clinical symptomatology. In addition, when you look at tangles biochemically, you find that they can have different structures in different diseases. There are different isoforms of tau that all come from the same gene, and one exon, which is alternatively spliced, can give rise to tau, for example, with either 3 or 4 microtubule binding domain repeats, and that's called 3R and 4R tau, and you can use that as a marker to differentiate tangle types in tauopathy. In addition, the filaments themselves can have different ultrastructure. You can have tangles that form paired helical filaments, which is the classic form that you see in Alzheimer's disease. You can also see straight tubules and twisted ribbons and all sorts of other things in other tauopathies. So what are these tauopathies, and when do we see tangles? There's a subset that we call the refer to as sometimes as the secondary tauopathies. For example, Alzheimer's disease, where amyloid is believed to be the primary cause of neurodegeneration, and the tangles are secondary. And there are other amyloidoses where you also see neurofibrillary tangles without the Alzheimer's amyloid that's composed of other proteins. The primary tauopathies is a group of diseases where tau is the main neuropathological feature, and there are a lot of them. We see neurofibrillary tangles in chronic traumatic encephalopathy, which is a neurodegenerative disease that follows repetitive mild traumatic brain injury, and I'm going to talk a little bit about that. There's also a host of other diseases. I'm going to talk a little bit about a new entity that I've been obsessed with for the past 7 or 8 years, and right now we're coming to the point where people have started to recognize that it may be its own distinct tauopathy, but there's also movement disorders, for example, progressive supranuclear palsy and corticobasal degeneration. There's globular tauopathy. Mutations in the tau gene themselves are also associated with neurodegenerative disease. These are coding region mutations or mutations in splice sites that affect the splicing of tau. We also see tangles in other kind of more exotic contexts, for example, myotonic dystrophy, which is associated with mutations in proteins that influence splicing, the lytico-bodig of Guam, the ALS Parkinsonism dementia complex, which surprisingly is associated with potentially toxin, although that's controversial. There are viral diseases of the nervous system that are associated with neurofibrillary tangles. Nobody really quite knows why, but in subacute sclerosing panencephalitis and post-encephalitic Parkinsonism, you can see tauopathy. And even in neoplastic context, for example, gangliogliomas, a subset of them will have neurofibrillary tangles. Among these sporadic tauopathies, they don't have mutations in the tau gene necessarily, but some of them are associated with common variation in the tau gene that seems to be serving as a risk allele. The frontotemporal dementias with tau mutation, I think, are important. They are important because they were the first cases that really established that tau dysfunction on its own is sufficient to cause neurodegeneration, that you don't need an amyloid or another factor to drive it, that when tau's not working, you're going to get tangles and neurodegeneration. So how does it work? What causes these tangles, and why are they killing neurons? Well, we don't really know, but if you look at these, we don't know for sure in all contexts, but if you look at these diseases, you can get some clues. We have genetic causes. We have the tau haplotypes, which I'll talk about a little bit more later. Tau deletion or duplication can influence disease. The mutations in the tau gene are important. But most of the patients that have tauopathy don't have mutations in tau. These are the primary tauopathies. But they do seem to have changes in splicing. So people are interested in alternative splicing of tau, and you can even have tau mutations that influence splicing that cause neurodegeneration. Somehow, tau gets hyperphosphorylated, and many people believe that addition of a phosphate to the tau protein will modify its function in a similar way that the mutations might modify the function of tau. So hyperphosphorylation or mutation of tau has been shown in some contexts to decrease the binding of tau to microtubules. It also increases its propensity to aggregate. So the relevance of that to neurodegeneration is of interest. In Alzheimer's disease, one hypothesis, although we don't really know exactly how amyloid causes tangles and neurodegeneration, but one idea is that amyloid toxicity triggers a kinase cascade that triggers hyperphosphorylation. One thing that's kind of interesting, just as an aside, is that we see hyperphosphorylation of tau in normal brain development, and also there have been reports that hibernating animals also can develop physiological phosphorylation or hyperphosphorylation of tau. Anyway, this tau protein will then aggregate, oligomerize, and then somehow neurons die. And how that happens, we're not sure, but neuroinflammation, oxidative stress might play a role. Autosomal autophagy failure or lysosomal failure is another hypothesis. And I'll just mention briefly that there's been a lot recently about the possibility that tau itself might function in a prion-like way, templating the conformational change of additional tau molecules and propagating. That conformation can propagate through, potentially propagate through the brain, just as a prion might. So the first tauopathy that I'm just going to talk about a little is chronic traumatic encephalopathy. So it's been known for close to 100 years that boxers can develop a severe encephalopathy. And this was described first by Harrison Martland, who was the Newark medical examiner, a very famous, well-renowned medical examiner. And he published a report. He called this encephalopathy, this pattern, punch drunk. And later this entity came to be known as dementia pugilistica, dementia of the fighter. And it was thought, and likely true, that the forces that are transmitted to the skull are then transmitted into the brain, which lead to angular, rotational forces that shear brain structures, and axons and blood vessels are vulnerable, of course. But it wasn't until many, many, many years later that Bennett Omalu, working in Pittsburgh, the medical examiner, did an autopsy on an American football player and noticed neurofibrillary tangles and also amyloid plaques that can be associated with chronic traumatic encephalopathy, and he made this link. And this triggered a massive effort to identify more cases of American football players who had this pattern, as well as other people who have been exposed to repetitive traumatic brain injury. We had known that you could see chronic traumatic encephalopathy in headbangers, in victims of domestic violence, and a lot of other contexts, but this was really new, that something as prevalent and important in our culture as football could be associated with this devastating neurological disease. One of the results of all of this interest has been these new criteria for chronic traumatic encephalopathy. So Anne McKee is leading the neuropathology effort up at Boston University, and I have had the privilege to participate in this study, as has Dennis Dixon, who's going to be speaking in a couple minutes. What Anne and her colleagues have done was to collect the largest cohort of elite athletes, football players, other athletes who participated in contact sports, as well as military veterans who've also had exposure to blast injury, and characterize the changes in the brain. And they proposed, and now it's validated, that there's a distinct neuropathological signature of chronic traumatic encephalopathy that can be distinguished from all of the other tauopathies. And it's based primarily on the regional distribution. So one of the things that we see is focal collections, patchy collections of tau in regions of stress concentration, places in the brain where it would be predicted that forces would concentrate. And so the depth of the sulcus is one of them. You can see here and here that there's this increased staining in these cerebral cortices stained with phospho-tau from patients with CTE. There's another pathognomonic lesion, which is the perivascular accumulation of phospho-tau within neurons and glia in the cortex. There is a pattern of phospho-tau accumulation in glia, which is in the white matter, which we're now referring to age-related tau astrogliopathy, which is distinct. These changes this pattern is very, very unique, and it's very easy to pick out if you have access to tau immunostaining and you've sampled the brain effectively. And the new criteria for CTE give guidelines for which brain regions are best to sample to look for CTE. And just to point out very briefly, what is this good for? Well, neuropathologists have started already to apply these new criteria to case series that exist. And of course, people are going to be applying the criteria to new cases in the future. But Dennis Dixon and his team have looked back in their brain bank, which is a large neurodegenerative disease series, and they found among autopsied brains from patients with an athletic history, they found a whopping 30% of them had signatures of chronic traumatic encephalopathy. And there have been a lot of other studies that have started to look at that. So this is really important. And I'm going to shift gears a little bit and talk about my main interest, which is related to Alzheimer's disease. So it's been known, let me go back. Alzheimer's disease, as I alluded to before, is defined by two histopathological features, the amyloid plaque and the neurofibrillary tangle. There have been many, many different schemes over the years to classify patients with Alzheimer's disease. Some of them are amyloid-based, some of them are tau-based. And over the years, these criteria have been revised to take into account more modern diagnostic approaches. In 2012, I'd like to point out that the criteria were revised, and the way that this worked, by the way, this whole project was a multi-institutional collaboration with the NIA and the Alzheimer's Association, and it was spearheaded by Dr. Montine, who's going to be speaking following me about Alzheimer's disease. And we're delighted to have him, and co-led by Brad Hyman at Harvard University. The way that they decided to do it was to take amyloid and tau and build a matrix using semi-quantitative and quantitative measures of these pathologies, and use that information to make probabilistic statements about the likelihood of whether or not somebody has Alzheimer's disease. There were other innovations that went along with these new criteria. One was to dissociate the clinical variables from the neuropathology. And that was because previous criteria mandated that you had to have a documented history of dementia in order to give a diagnosis of Alzheimer's disease. And many neuropathologists don't have access to that clinical information. So we now talk about Alzheimer's disease neuropathological change independently of the clinical. But there was this group that there was a lot of controversy about and a lot of discussion and no agreement. These are patients that develop Alzheimer's type tangles in the absence of amyloid plaques. Now these patients, they weren't quite sure how to handle them. They did suggest that these patients that don't have plaques are not Alzheimer's disease. Although there was disagreement, there was less agreement here at the higher end of the scale. And so while this was happening, I was at Columbia, and I became aware of an entity that was better known among neuropathologists, not so much outside of our field, of something called tangle predominant dementia or tangle-only dementia. And this was first described in 1992 by Ulrich et al. In a large autopsy series in Europe, where they found 10 cases, predominantly women, who developed massive numbers of neurofibrillary tangles localized to the medial temporal lobe. So this is an early to moderate stage of Alzheimer's disease, not the high BRAC stage where you have diffuse tangles. The women were mostly demented with amnestic features. Tangles were not present in the frontal parietal occipital cortex. And what was striking was that there wasn't a whiff of amyloid anywhere in the brain, or no plaques, or very, very, very scarce plaques. And they weren't really quite sure what these patients represented. And this was quickly replicated by Kurt Jellinger in Europe, and really nobody followed up on it. There were a couple of case reports and some studies in Japan, but this is what they look like. This is a brain from the Columbia University autopsy series, courtesy of Jean-Paul van Sadel. This is a hemi-section through the cortex. You can see that this medial temporal lobe is shrunken and atrophic. There's dilatation of the lateral ventricle. The cortex is relatively preserved. And when you do your histology, you find that the hippocampus is positive for massive amounts of phospho-tau, just like you see in Alzheimer's disease. In fact, you universally see neurofibrillary tangles in the medial temporal lobe in people over about the age of 50 in the context of normal cognition. But these patients are demented, and when you look at your amyloid, there isn't a whiff of it anywhere. This is an immunohistochemical stain for amyloid. The Alzheimer's cases will have a lot. Even the control patients will often have amyloid plaques. So what is this? And it seemed to be different, and I became kind of obsessed with it, because the possibility that people could be developing Alzheimer's disease without any amyloid ostensibly flips the whole hypothesis that amyloid is causing disease on its head. This is what they look like when you look at high power. You see all of these tangles. When you do a silver stain, you see these classic flame-shaped tangles, lots of these ghost tangles, which are the necrotic remnant of a neurofibrillary tangle, and the insoluble, protease-resistant paired helical filaments remain. This is a ghost tangle. You see lots and lots of these dead cells, the ghost tombstone tangles, sometimes they call them. When you stain for 3 repeat tau and 4 repeat tau, you see that they're positive for both. And this is the isoform pattern that you see classically in Alzheimer's disease. So they look like Alzheimer's disease. And when you do your electron microscopy, they have these paired helical filaments, just like you see in classic Alzheimer's disease. So these really look, for all the world, like Alzheimer's disease type neurofibrillary tangles, yet there's no amyloid. And what does that mean? There are quite a few patients or subjects that have this pattern. This is unsupervised hierarchical clustering of neuropathological variables, semi-quantitative counts of pathology and neuron loss. And there is a group here that clearly segregates away from the classic Alzheimer's cases, which have amyloid. And so this group is what we've been looking at. And so it kind of was a paradox, right? Because some people told me that, this is Alzheimer's disease. It's really a soluble amyloid oligomer, which is the cause, not the plaque, and or subtype of Alzheimer's disease. And what you need to do is go look biochemically, because neuropathology is not good enough for detecting those types of changes. And so I considered that idea. We also looked closely at the tau gene. When I was trying to find cases like this, people told me, do sequence tau because you'll probably find a tau mutation. So we looked at tau. Another thought was, when we started writing papers, that the reviewers came back and the grant reviewers came back and said, you're just looking at something that's normal because you see tangles in that same distribution. You're comparing normal to normal, and you're not going to find anything. And so I got a little frustrated about that. But the idea was, well, maybe it is normal or some kind of pathological aging. Maybe it's something new. Or, I showed you that list of other tauopathies. It could be a viral thing, traumatic or toxic. So what is this? So we considered the amyloid hypothesis. So this has really guided Alzheimer's disease research. The idea is that amyloid, that the A-beta peptide that makes the plaque, increases in that are the primary driver for the disease. And everything else, tangles, neurodegeneration, is secondary. And there's a lot of evidence to support that. And it comes from kindreds that have mutations in the amyloid precursor protein and other genes that increase the amount of amyloid, and it's an autosomal dominant pattern, and you variably get the disease. That genetic evidence is very strong. However, there were smart people out there that disagreed, and one of the criticisms was, well, amyloid plaques don't correlate very well with dementia or neurodegeneration. In fact, tangles correlate a lot better. And another thing that was curious is that there's this spatiotemporal disconnect where you actually get tangles first before you get plaques. And they occur in different parts of the brain. The tangles start to appear at the bottom of the brain in the medial temporal lobe, and your plaques appear in the neocortex at the top. So what is that? What's the explanation for that spatiotemporal disconnect? And there was also a lot of anxiety when I started because there were several large phase 3 clinical trials targeting amyloid that had failed. There were answers, possible ideas about why this might be the case, but we wanted to look at the tissue. So the first thing I did was, with my postdoc Ismael Santa Maria, we got as many cases as we could find and started doing biochemical measurements of the amyloid. And instead of finding increased soluble oligomers of amyloid, we found virtually undetectable levels of A-beta 1 to 40 and A-beta 1 to 42 in the parenchyma of these cases with no plaques. And that seemed to make sense because they didn't have any plaques. They had very low levels, and lower even than the average control, and far, far lower than what you see in Alzheimer's disease. And we also looked at the processing of the amyloid precursor protein, and it looks like in a subset of these patients, they are shuttling their amyloid down the non-amyloidogenic pathway. They basically have enzymes that are cleaving it in a way that prevents amyloid from getting formed. We also started to look at the genetics, and there's no association with the ApoE allele, which is the most common risk factor for sporadic Alzheimer's disease. So it looked like, and that ApoE epsilon 4 allele is strongly associated with amyloid deposition. So the idea was that, well, maybe they have changes that protect them from amyloid, genetic and biochemical changes protect them from amyloid plaque formation. And it was hard to argue that this was an A-beta dependent mechanism driving the disease. Is this a variant of FTLD? So I mentioned before that there are mutations in tau that can influence the splicing and mutations that are in coding regions that can influence binding to microtubules. So as I mentioned before, in this paper, Mike Hutton's paper, there's a lot of people involved with this at the Mayo Clinic. They showed that there are causative mutations that are independently associated with neurodegeneration in the absence of amyloid plaque formation. So we asked, do these patients have mutations? And alternatively, do they have differences in their splicing when we do this molecularly? And just to summarize this quickly, they did not have a mutation in the tau gene that we could find. They form neurofibrillary tangles that have the same phosphoprotein hyperphosphorylation pattern that you see in Alzheimer's disease, and the isoforms are essentially identical. When you purify tau out of these brains, you see once again those same paired helical filaments, just like you see in Alzheimer's disease. So we thought it was hard to argue that this pattern is FTLD. So then there's this idea, well, maybe this is normal. And so as I mentioned, neurofibrillary tangles are universal in aging in the temporal lobe, and that was first shown by Patrick Hoff and others here at Mount Sinai. What differentiates these cases is that they have dementia, and they have a lot of neurofibrillary tangles, but they seem to be localized. So as we were looking for cases, we stumbled across this model, the Rowe and Kahn model. And so for the students in the audience who are interested in aging, and in all forms of aging, this is required reading. So I put the reference down here at the bottom. What Rowe and Kahn did was they looked at a lot of studies across a lot of organs, a lot of contexts, and they pointed out that normal is a problem. The way you define normal in aging, the way we usually do it is we say, well, you failed to meet the criteria for a specific disease, so therefore this is a normal control. But if you look at those normals, you see a lot of variability. And what they did was they said, split up your normals into usual aging and successful aging. And they pointed out, for example, if you have a 90-year-old whose memory is shot, you might consider them to be normal when you compare them to age-adjusted controls. But if you compare them to their younger selves, they're going to be a shadow from that. So the usual aging is that which is kind of commonly encountered, that kind of normal. And then there's another kind of normal, which is your successful aging, where when you're compared to your younger counterparts, you're still doing great. And it turns out that these groups are important, and they're different. The usual aging is dependent across a lot of different studies, more on extrinsic factors. Whether you drink, whether you smoke, if you want to be, if you don't want to have usual aging, you can adjust your behavior. So that's actually a very positive finding. And Jack Rowe became very famous, wrote this book, Successful Aging, made a ton of money, and what have you. But the problem is that these successful agers, intrinsic factors, mostly genetics, kind of dominate. So if you want to make it to 100, pick your parents very well. That's the idea there. So we started to think about these tangles in the temporal lobe, not as a disease entity, but as a kind of age-related quantitative trait. And we started breaking up our groups into usual aging and successful aging, and we started to see different things. And so the idea is that instead of saying disease, not disease, we break them up, mild, moderate, and severe. These severe cases are tangle-only dementia, and then we have our kind of usual agers here with some tangles, and then the people who are tangle-free at 90 that are successful agers. And then when you look at your tangle staging, you can see that there's a lot of variability in the regions. There's a lot of variability in tangle burden in the hippocampus in the absence of amyloid. And so this is our kind of new model where we hypothesize that your NFT burden correlates with your amnestic or your cognitive impairment, and the tip of the iceberg are these tangle-only dementia cases, and that there's a lot of people with what we call mild cognitive impairment or age-associated memory impairment that have intermediate levels. And when we did our genetics, we found something different that we didn't see before. I'll have to just take a second to show you guys that the tau gene is really interesting because it sits in an unstable chromosomal locus. There are low copy repeats on each side of it, and they recombine through non-homologous recombination and flip the gene. And this actually has given rise to two haplotypes that they call H1 and H2. There are no mutations in the tau gene. It's just the orientation and these kind of accumulations of additional duplications on each side. And this has actually happened not once, but many times during primate evolution, and has led to these haplotypes being so different that they can never recombine. They're in complete linkage disequilibrium. This pattern was first discovered in progressive supranuclear palsy. Other tauopathies seem to have this. A big surprise is that this this haplotype is associated with Parkinson's disease as well, which is not a tauopathy. It's associated with alpha-synuclein deposition in Lewy bodies. That was a big surprise. The link to Alzheimer's disease is controversial. There is a subgroup. Okay, there's a subgroup of ApoE4 negative Alzheimer's patients that seem to have maybe an association with H1. So we found that these age-related tangles are on a continuum, so to speak, with these different categories with respect to their frequency of the H1 haplotype. So just kind of one last little hypothesis here. We don't know what causes this pattern, but if you look at chronic traumatic encephalopathy, you'll notice that there is a massive temporal lobe, a tau, regional tau deposition that parallels what you see in this age-related tauopathy. This is an 80-year-old woman with mild dementia. She didn't play football, and this is a 56-year-old NFL player, and he has this patchy tau deposition in the neocortex, but he also has a fried temporal lobe. So one of the things that we don't know is the cause of the trigger, but I don't think it's too early to start to speculate. Okay, and so let me just leave you with this idea. This is not my idea. This came from my mentor, Dr. von Sadel, who came up with it with E.P. Richardson. They thought that maybe these age-related tangles that they were seeing were a form of traumatic brain injury, not mild TBI like the concussions that we're seeing in CTE, but maybe a very mild sort of microtrauma that accumulates throughout life, a kind of wear and tear type of traumatic brain injury triggered by foci of stress concentration at the base of the brain where the hippocampus abuts the tentorium. So that's where we are. So now we have new criteria, and you can find that if you go to the web. And when you use those criteria, it's associated with cognitive impairment, and it probably has some relevance for biomarkers because they're finding lots of people out there who have positive tau in the cerebrospinal fluid, but no amyloid. And in the future, we're going to be collecting large numbers of these cases and doing a large genetic study collaboratively with over 25 different centers. And these are the people to thank. People in my lab, predominantly Ismael Santamaria, who now is an assistant professor, tenure track at Columbia University after he left. So anybody who wants to work with me, that's what they could potentially look forward to. Thank you very much.